So normally, vaccine development feels like this totally exhausting game of molecular whack-a-mole.
Oh, absolutely.
You know, a virus mutates, it pops its head up, and we just scramble to develop a vaccine to hit it. And by the time we actually do, the virus has already mutated again and popped up somewhere else.
Right. It's a constant catch-up game.
Exactly. But what if an artificial intelligence could help us win the game before the virus even strikes? Because today, for you listening, we are going on a deep dive into a really major breakthrough out of the University of Cambridge and their spin-out company, Diasynvax.
Yeah, it's fascinating stuff.
It really is. We're looking at the June 2026 Phase 1 trial results for the very first vaccine where the key antigen was, well, entirely designed by AI.
And I think the overarching mission here for us today is to really understand how this specific technology shifts humanity away from just, constantly chasing mutating viruses.
Right.
And moves us toward actually anticipating them, because they aren't just trying to stop one specific variant of one specific virus. The objective here is to proactively build a shield against entire families of pathogens.
Including things like zoonotic threats, right?
Yeah, exactly. Like bat viruses that haven't even made the jump to humans yet.
Which is wild. And to really grasp how massive that shift is, we kind of have to look at why we've been stuck in this reactive loop for so long.
Yeah.
I mean, the past century of vaccines, and especially, the mRNA vaccines deployed during the COVID-19 pandemic.
Which were huge triumphs.
Total masterclasses and rapid scientific response, absolutely. But they were still fundamentally a reaction to an event that had already happened.
Yeah, and that limitation really comes down to the underlying biology of RNA viruses.
Okay, unpack that for us a bit.
So RNA viruses, which include coronaviruses as well as influenza, they're incredibly unstable at a molecular level.
Like, they change shape.
Basically, yeah. When they replicate inside a host, they lack the robust proofreading mechanisms that our own human DNA replication has. So they constantly make these tiny spelling errors in their genetic code.
Just typos in the code.
Right, molecular typos. And over millions of replications, those genetic errors physically change the three-dimensional shape of the virus, particularly the proteins on its surface.
Got it.
And traditional vaccines, and even mRNA vaccines, they are essentially designed to train your immune system to recognize today's specific viral shape.
So tomorrow, when the virus mutates and changes that shape, the vaccine kind of loses its efficacy.
Exactly.
The wanted poster is suddenly out of date.
That's a great way to put it.
So enter the team at Cambridge and Diasynvax. They've been working on this since, 2017, utilizing a concept called reverse vaccinology.
Yes.
And now this isn't just a slight upgrade to the old method, right? It completely upends how researchers find the target in the first place.
It really does, because historically, scientists would isolate the physical virus in a laboratory, break it apart, and run extensive physical tests to see, which chunk of it triggers the strongest human immune response.
Trial and error, basically.
Very much so. But reverse vaccinology bypasses the physical virus entirely. You start with the pure genetic code.
Okay.
What Diasynvax did was feed their machine learning algorithms massive amounts of genetic sequence data, stuff gathered from global coronavirus surveillance programs.
So instead of looking at the virus under a microscope, they are feeding an AI millions of, architectural blueprints.
Yes, exactly.
They're essentially asking the AI to look at every single coronavirus building in the city and find the one structural pillar that supports all of them.
That is an excellent way to visualize the mechanism. The AI ingested the genetic instruction manuals not just for SARS-CoV-2, but for the entire sarbecovirus family.
That's a pretty broad family, right?
It's very broad. That family tree includes the original SARS virus that caused the 2003 outbreak, the various strains of COVID-19, and a vast spectrum of bat viruses currently circulating in animal reservoirs.
Wow. All of that data.
Yeah, and the algorithm analyzed all these sequences specifically to identify what we call conserved features.
Meaning the parts of the virus that physically cannot change without destroying the virus itself.
Precisely. The essential load-bearing walls of the virus, to use your analogy.
Right.
If the virus mutates those specific genetic instructions, it basically loses its ability to function and infect human cells.
It falls apart.
Exactly. So the AI mapped those common unchangeable features across the entire viral family, and then it used that data to computationally design a super antigen.
A super antigen.
Yeah, which is a completely synthetic molecular blueprint. It's designed to train the human immune system to recognize those permanent structural pillars regardless of how much the rest of the virus mutates its outer coat.
Okay, let me push back on the AI element for a second, though. Because we've been mapping genomes for a long time. Human scientists are incredibly smart, and we have massive databases. Why couldn't a team of researchers just cross-reference these genomes and find those conserved features on their own?
It's a fair question.
Like, is the AI really doing something novel here, or is it just acting like a super fast spreadsheet?
Oh, it is much more profound than simple processing speed. It really comes down to the sheer multidimensional complexity of protein folding.
Okay, how so?
Well, a linear sequence of genetic code translates into a long chain of amino acids. That chain then folds into a highly complex three-dimensional physical shape.
Right, biology is 3D.
Exactly. A human scientist can easily compare genetic sequences and spot linear similarities on a screen.
Like writing text.
Right. But predicting how millions of different synthetic amino acid combinations will fold in three-dimensional space, and then predicting exactly which of those microscopic shapes will optimally bind to human immune receptors.
That's a lot of variables.
It requires analyzing thousands of interconnected variables simultaneously. Human cognition processes information sequentially.
One step at a time.
Yeah. But deep learning neural networks process these vast, multidimensional patterns in parallel. The AI can essentially see structural relationships across thousands of genomes that our brains simply aren't wired to visualize all at once.
Okay, that makes sense.
Yeah.
But knowing these conserved features in a computer simulation doesn't actually solve the biological problem.
No, it doesn't.
I mean, a perfect digital blueprint is completely useless if it falls apart the second you put it into a human body.
Yeah.
So let's look at the June 2026 Phase 1 trial results published in the Journal of Infection.
Let's do it. The trial evaluated this AI-designed vaccine, which they designated as PEACPS.
Catchy name.
Very catchy. They tested it on a cohort of 39 healthy adults ranging in age from 18 to 50. But, the AI antigen design isn't the only revolutionary aspect of this trial.
Right, because we have to look at the physical delivery mechanism. This vaccine was administered without a needle.
Which is fantastic news for anyone who breaks into a cold sweat at a doctor's office.
Absolutely. But how exactly does that work physically?
Oh,
I mean,
okay.
you can't just splash liquid on someone's arm and hope it gets absorbed into the bloodstream.
No, definitely not. It was delivered using a microfluidic jet.
It's a jet.
Yeah, instead of a solid metal needle physically piercing the tissue, a specialized device forces a microscopic stream of liquid at an incredibly high velocity.
Oh, wow.
So the fluid itself becomes the needle. It parts the cells and penetrates the skin painlessly, depositing the vaccine directly into the intradermal layer.
That sounds like science fiction. Is there a medical reason to do it that way besides just avoiding needles?
There is, actually. This is a major biological advantage because the dermal layer of skin just beneath the surface is densely packed with specialized immune cells.
Okay.
So it triggers a highly efficient immune response compared to a deep muscle injection. Plus, as a practical clinical benefit, eliminating the visual trigger of a hypodermic needle drastically reduces cases of vasovagal syncope.
Which is people passing out from seeing the needle.
Exactly, anxiety-induced fainting.
So we have a high-tech delivery method. But the payload itself is also entirely different from the major COVID-19 vaccines most people remember receiving.
Yes, those were mRNA vaccines.
Right, and this new AI-designed one is a plasmid-based DNA vaccine. What exactly is a plasmid, and why does making it out of DNA even matter?
Think of a plasmid as a tiny, highly durable, circular ring of DNA. It basically acts as a microscopic delivery vehicle carrying the AI's superantigen instructions into your cells.
And why is DNA better than mRNA here?
The shift from mRNA to DNA is a massive game changer for global logistics. Messenger RNA is a single-stranded molecule.
Okay.
Because it's single-stranded, it is inherently unstable, and the molecular bonds break down very quickly unless the vaccine is kept at ultra-cold temperatures.
Right, I remember all those stories during the pandemic about the specialized freezers.
Yeah, requiring minus 80 degrees Celsius freezers just to move the vials from point A to point B. It was a logistical headache.
And DNA is different.
DNA is a double helix. The two strands bind together, protecting the genetic information in the center. So it is structurally far more robust.
Meaning it doesn't require a deep freeze to survive the trip from the manufacturing lab to the patient.
Exactly. It remains incredibly stable at standard refrigeration temperatures, and often even at room temperature.
Wow.
That means you can transport this vaccine to remote regions and developing nations that completely lack ultra-cold chain infrastructure.
Okay, so the underlying science sounds pretty bulletproof. But let's talk about what actually happened when they gave this DNA ring to those 39 people.
Right, the real world test.
First, the incredibly good news. The trial data showed the vaccine was extremely safe and well tolerated. There were no serious adverse effects.
Which is always step one.
But, however, the resulting immune response was categorized by the researchers as modest but variable.
Yes.
It did successfully generate an immune response against SARS-CoV-2, the original SARS, and related bat viruses. But the variation is what catches my eye.
It's an important point.
If the AI designed the absolute flawless antigen in a simulation, why was the human response variable? Did the algorithm, make a mistake?
The algorithm didn't make a mistake. What we are seeing here is the profound friction between a pristine mathematical algorithm and the chaotic reality of human biology.
Chaotic is a good word for it.
Right. In a computer simulation, or even in a sterile lab testing genetically identical mice, you have a blank slate. You get clean, uniform data. But when you move to 39 living, breathing human beings in the year 2026, you are dealing with an immense amount of biological noise.
Biological noise. What does that actually look like inside the immune system of a trial participant?
Well, it looks like a deeply complex immune history. Every single adult in that trial has previously been exposed to COVID-19 or has received multiple doses of various COVID vaccines over the past several years.
Their immune systems are not blank slates.
Not at all. They have already been heavily trained and shaped by those prior exposures, and there's a well-documented biological phenomenon known as immune imprinting. When the immune system sees a new vaccine that looks somewhat similar to a virus it already knows, it tends to rely on its older, existing cellular memories rather than perfectly learning the new instructions.
Oh, okay.
So when the AI's new superantigen enters the body, the immune system's prior baggage complicates the reaction.
So if you're listening to this and thinking, wait, I've had COVID twice and got 3 boosters, will this AI vaccine even work on me? This is the exact biological hurdle the researchers are dealing with.
Exactly.
The AI handed us a perfectly clean, brand new blueprint, but our immune systems are like a heavily marked up chalkboard. The body is struggling to read the new instructions over the old erasing smudges.
That is an incredibly accurate way to frame it. And yet, despite all of that biological noise and immune imprinting, the vaccines still managed to generate cross-reactive responses across the sarbecovirus family,
Even the bad viruses.
including those bat viruses that haven't even jumped to humans yet.
That's amazing.
It is. That is why experts in the field, including Professor Saul Faust and Andy Pollard, are emphasizing the massive potential of these early-stage results.
So what comes next?
The immediate next step is a phase 2 trial involving around 200 people. That larger cohort will provide the statistical power needed to filter through the noise, adjust the dosing, and truly map the vaccine's efficacy.
And this approach isn't just trapped in the world of coronaviruses either. Building on the messiness of human biology, we have to look at the massive potential on the horizon.
It's expanding rapidly.
Yeah, the team at Cambridge is taking this exact same reverse vaccinology process and pointing the AI and other major global threats. They are currently working on a universal vaccine for influenza.
Which is huge.
Think about what that means. Instead of the endless guesswork of the annual flu shot, where scientists trying to predict which strain will be dominant next winter, an AI finds the unchangeable pillars of the flu virus.
You could potentially get one shot that protects you for a decade.
Exactly. And they are also actively applying this to Ebola. Professor Jonathan Heaney, who leads the Diosynvax team, he noted that it is truly incredible what they can do with this technology for the good of humanity.
The potential is definitely staggering. But to move from a phase 1 trial to a globally deployed, future-proof vaccine, we have to look soberly at the real-world hurdles still standing in the way.
Right, it's not a magic wand.
No. Beyond the scientific hurdle of the upcoming phase 2 trials, there are massive technical challenges. Scalability, for instance.
How to make enough of it.
Exactly. Manufacturing microfluidic jets and plasmid DNA for a clinical trial of 39 people is one thing. Scaling that highly specialized manufacturing pipeline to produce billions of doses requires an entirely new industrial infrastructure.
And then there's the hurdle of getting the regulatory agencies on board. This brings up something regulators call the black box problem, doesn't it?
Oh, absolutely. When a pharmaceutical company submits a traditional new drug to the FDA or the EMA, the regulators want to see the exact mechanism of action.
They want the receipts.
They want a human scientist to explain, step by step, exactly why they chose a specific molecule and exactly how it interacts with the body. But deep learning neural networks don't output their internal reasoning.
Right, they just give you the answer.
Exactly. That is a profound challenge for regulatory science. The algorithm ingests massive data sets and outputs a brilliant, highly effective molecular design. But the computational pathway it took to arrive at that design involves billions of microscopic, dynamic weightings across a neural network.
It's just a bunch of math that we can't untangle.
It is mathematically impossible for a human being to fully reverse engineer the AI's internal logic. So we are asking regulatory health agencies to approve a biological intervention designed by an intelligence whose exact deductive process we cannot fully explain.
Which creates an enormous communication problem for the public.
Definitely.
Because if top-tier scientists and PhD-level regulators are struggling with the black box nature of the AI, how on earth do we convince the general public to roll up their sleeves?
It's a tough sell.
Think about the messaging. Take a needle-free blast of AI-designed synthetic DNA. That sounds incredibly intimidating. Doesn't this require a massive fundamental upgrade in how we communicate science?
It absolutely does. Earning public trust in science is a steep mountain to climb. The solution relies entirely on rigorous, transparent clinical evidence.
They have to show the proof.
Right. We might not be able to perfectly map the AI's internal thoughts, but we can completely verify the AI's physical results. Phase 1, Phase 2, and Phase 3 double-blind human trials are the ultimate proof of safety and efficacy.
The data has to do the talking.
The biological data has to speak louder than the algorithm, and practically speaking, we also have to solve the logistical distribution hurdles. A vaccine designed to anticipate and stop a bat virus spillover is completely useless if it doesn't physically reach the remote populations living in proximity to those animal reservoirs.
If the shield can't get there, the shield fails. But if we can navigate the black box of regulation and the public trust and the global distribution logistics, the future opportunities extend way beyond just viruses.
They really do.
The sources mention that this computational approach could be applied to solving antibiotic-resistant bacteria, finding the conserved features in mutating superbugs. They're even looking at developing therapeutic cancer vaccines.
Because cancer is fundamentally an evolutionary problem, much like a rapidly mutating virus.
Really?
Yeah, cancer cells mutate quickly to evade the body's immune system. If an AI can analyze the specific genetic profile of a patient's tumor, identify the conserved features of those cancer cells, and design a bespoke superantigen to train the patient's own immune system to hunt them down.
Wow.
We are looking at a completely new paradigm for personalized medicine.
It really feels like we are standing on the edge of a new frontier here.
Yeah.
So let's synthesize what we've learned in our discussion today.
Sounds good.
The June 2026 results from Cambridge and Diacinvax prove that artificial intelligence has officially matured. It is no longer just a high-speed data cruncher that helps humans do their jobs a little faster.
Not anymore.
It has become a true co-creator in biomedicine. It is actively merging computational biology with viral evolution to make humanity fundamentally more resilient.
We are witnessing the transition from reactive medicine to proactive, future-proof health.
Which is a massive shift.
The old model was waiting for a pandemic to start, suffering the devastating consequences, and then racing to build a defense. The new model is building the defense against the entire family of viruses before the first human is ever infected.
Which means the era of playing molecular whack-a-mole might actually be coming to an end.
We can certainly hope so.
But before we wrap up, I want to leave you, the listener, with a final provocative thought to chew on, something that builds on everything we've talked about.
I love these.
We've spent this entire time talking about using AI to perfectly predict and counter the natural evolution of external threats, animal viruses, pathogens, things outside of us. But what happens when we turn those algorithms inward?
Oh, wow.
If AI can design a super antigen to train our immune system to fight a bad virus, will the vaccines of the future actively anticipate and rewrite how our own immune systems handle internal failures?
That's a huge question.
Could an AI predict how your specific immune system will degrade as you age and design a synthetic DNA update to prevent autoimmune diseases before they even trigger? Are we just looking at the end of pandemics, or are we actually looking at the beginning of programmed human immunity?
It is a staggering possibility, and it forces us to rethink the very limits of our biological destiny.
Because suddenly, the muddy waters of human biology might just become as clean and decipherable as the code that's trying to save it. Thank you so much for joining us on this deep dive into the first AI-designed vaccine. Keep questioning, keep exploring, and stay insanely curious.